Hello and welcome to my video on digestion and enzymes for GCSE. The things we're going to be covering in this video are the properties of enzymes, the biological molecules within the diet, and the digestive system organs. Enzymes are protein molecules which act as biological catalysts. This means they speed up the rate of chemical reactions without being used in living organisms. There's the lock and key model, which gives an idea of how enzymes and their substrate fit together. The enzyme is a protein produced by the organism, whereas the substrate is a molecule which undergoes a chemical reaction. Okay, on the diagram to show the enzyme action, you can see the active site. This area has what's called a complementary shape to the substrate, that is, it looks like a mirror image of it, so the two can fit together neatly. For the enzyme and the substrate to react, they need to collide. This means that at low temperatures the rate of reaction is lower as there's less collisions. However, when the enzyme and the substrate do collide together, the enzyme then very slightly changes its shape. This causes a reaction to happen, which can break apart the substrate, forming two new molecules. This is what happens with digestive enzymes, as they break apart large insoluble molecules which couldn't be absorbed to make small soluble molecules which can be absorbed. This is a process that's called digestion. Because of the shape of the active site, enzymes are very specific, so each substrate has a single enzyme which can break it apart. Enzymes are also very sensitive to changes in pH. They have an optimum pH window where they work at their most efficient. When you get either side of this, either too acid or too alkali for them, the rate of reaction decreases sharply as the enzyme's denatured. When this happens, a protein becomes permanently scrambled, changing the shape of the active site and stopping the enzyme from working. The first of the molecules in a diet we're going to look at is carbohydrates. An example of a complex carbohydrate is starch, which is stored in plants. Animals store glycogen, which is a similar molecule. There are three simple sugars. The most common that you come across is glucose. Simple sugars are known as monomers as they can be put together forming repeating units of a larger molecule. Starch and glycogen are both examples of polymers. They're very large molecules made of repeating units of the simple sugars joined together. During digestion, carbohydrate enzymes are able to break apart the large polymers of carbohydrates. This results in simple sugars being produced. These are soluble, so they can be absorbed. The next molecule we're going to look at is proteins. Proteins are again polymers. They're made of long chains of amino acids which are joined together. There are thousands of different proteins, and these are all made by different combinations of the same 20 amino acids. So the monomer of a protein is a single amino acid. During digestion, you've got protease enzymes which break down proteins into the individual amino acids. Proteins are large insoluble molecules, so you couldn't absorb them, whereas the amino acids are small and soluble, so they can be taken up into the blood and the intestines. The third type of molecule which contains energy in a diet is lipids. Lipids aren't polymers because there's no single repeating unit. Their molecules have a single glycerol, and that's joined to three fatty acid tails which make up the lipid. The group of lipids contains fats and oils, and they're broken down by lipase enzymes. Lipids can't be absorbed, but the fatty acids and glycerol which are produced in this reaction can be taken up by the intestines again. Now to finish off, we'll put the process together and look at the whole of the digestive system. When food is first ingested, there's mechanical breakdown which takes place. That uses the mouth and the teeth, and it breaks the food into what's called a bolus. This is a small ball of chopped up food which is mixed with saliva. The bolus is then swallowed, moves down the esophagus to the stomach, and in the stomach and the small intestine you get enzymatic digestion. This is where enzymes are used to break down large molecules into small soluble ones which can be absorbed. There's pH changes which take place as food moves through the digestive system. Saliva in the mouth has a pH of around 7 to 7.5. As you move down into the stomach contains hydrochloric acid which has a very low pH of 1 or 2. The purpose of this is to kill pathogens, that's microorganisms which cause disease. As you move down into the small intestine, alkaline bile is produced by the liver and stored in a gallbladder. That serves to neutralise the pH of the stomach acid, creating optimum conditions for the enzymes present in the small intestine. Throughout the whole of the digestive system you have this process called peristalsis. What this means is the walls of the digestive system contain muscles. These muscles contract to squeeze the food along the digestive system to keep things moving in the right direction. These muscle contractions are so powerful that you can still digest your food properly upside down. Thank you very much for watching, and please join me again next time.